Good morning, everyone. So first of all, I'd like to thank the organizing committee for arranging this nice symposium and inviting me to come and speak here. I'm going to be talking about isolating the information conveyed by feedback connections in monkey and mouse visual cortex. So I'm aware it's organization for human brain mapping, but hopefully during the course of this talk, I'll make it apparent how this relates to human brain imaging. So my particular interest is in the function of feedback connections in the brain. So we know that information travels in two main directions in the visual system. So here we see on the slide a macaque-monkey brain. And feed-forward connections in the visual system travel from the retina through the thalamus into the primary visual cortex at the back of the brain. And then from there forwards into the higher visual areas, such as areas like V4 and IT. And these feed-forward connections arrive in layer 4, and they are strong driving connections which cause their target neurons to fire. Not only that, they define the functional properties of these target neurons. So they tell a neuron where it should respond to in space and what sort of features it should respond to, be that an oriented line in V1 or perhaps a particular face in IT. But there are just as many connections going in the opposite direction, so from the higher visual areas back down to lower visual areas. And it is these feedback connections which have a much more mysterious function, so we don't know exactly what they're doing. But I think most people would agree that feedback connections, well, at least part of their function, is to convey information about the brain's internal perceptual model of the world. But exactly which information is conveyed and how feedback affects ongoing activity in early visual areas is still a matter of intense debate in the field. Now, one of the reasons it's so hard to figure out what feedback is doing is because it arrives very early in early visual areas, and it does interact very strongly with feed-forward activity. And so a trick that researchers have used for many years is to actually isolate feedback from the feed-forward effects by using occlusion. So on this slide, we have a very famous image, the front cover of the Abbey Road album by the Beatles. And you might be able to see a subtle occluder that I have added to the image. So this large gray rectangle. And you could probably know who I've occluded and why I occluded him. So the trick here really is that by occluding part of the image, you have removed the feed-forward input from that part of the visual scene. There is no change in retinal activity in the occluded region of the scene. However, therefore, if we see any activity changes in that region, that these must be coming from other sources, from horizontal connections or from perhaps feedback connections. Okay, now, by then looking at what happens in this occluded region, we can therefore focus in on the function of these feedback connections and what sort of information they are bringing back to the early visual areas. And we can ask two interesting related questions. So first of all, are the feedback connections bringing back information about what would be predicted to be there? What is behind the occluder? Or in other words, do we get any Paul McCartney information in this particular image coming into the occluded region of the scene? The second related question is there any information in the occluded region of the image about the rest of the image? So would you get any information about George or Ringo in the occluded part of the image? And by answering these questions, we can help to try and understand what feedback is bringing back to the early visual areas and what possible function it might have. So I'm going to start with this second question. What information is there in the occluded region about the rest of the visual scene? Now, Lars Muckli, who introduced the session, has for many years used this occlusion paradigm in humans using fMRI. So in this study, they're typically using a natural scene, one of maybe 3 natural scenes, and the bottom right corner of the scene is occluded. And it's been shown many, many times that you can easily decode the information present in the rest of the image from the voxels that are in the occluded region of the scene. And this particular result comes from the 2015 paper where Lars did this in a laminar resolved fashion. So here the important result is the green line, where they showed that decoders which are built in different layers of cortex are able to decode the activity in the rest of the scene. And in particular, decoders in the superficial layers of cortex were the best at decoding which image was presented to the subjects. And these are the layers which receive mostly feedback connections in the visual system. Okay, so it does therefore seem in bold MRI that you can decode what is in the rest of the scene from the occluded region. But of course, there's the caveat here. This is bold. So bold doesn't necessarily correlate with the underlying spiking activity. And furthermore, we know that the bold signal is very sensitive to top-down connections. So if I was to actually put an electrode into V1 and look at the spiking activity there, would I still see responses to these occluded regions? And would we be able to decode the rest of the image from the occluded spiking activity. So that's exactly what we did. So we used an occlusion paradigm in monkeys. We did exactly the same sort of paradigm that we used in the humans with Lars's work. So in fact, we used exactly the same images that they used in this study in 2019 by Tyler Morgan. So Tyler kindly provided these images to us. These were 24 natural scenes, things like beaches, forests, but also things like industrial scenes and buildings. Now we showed these either non-occluded or with the bottom right corner occluded. And this was a very easy task for the monkeys. They just had to fixate on a fixation point, and then we showed the stimulus for 700 milliseconds. We recorded multi-unit activity with chronically implanted UTAR arrays in V1, and this was recording multi-unit spiking activity. And we were very careful to select only receptive fields which were in the bottom right quadrant of the image, and only those which were at least 2 degrees away from the boundary between the occluded and non-occluded region. So this is the response you get to these occluded and non-occluded images. Now this is not decoding or anything, this is just the basic spiking response of the cells. And what you can see first of all in blue are the response to the non-occluded images. And each line here is a response to one of these 24 images across cells. And what you can see is very, very rapidly, the responses diverge across these 24 images. So we can actually measure the time at which the responses to different images become different to one another, and that happens at around 46 milliseconds. Right, that's not too surprising here. There are different visual features in the receptive field for these different images, so of course the responses will be different. What's more interesting is for the occluded images. There, first of all, what we see is actually a rather weak response, right? So certainly the response is nowhere near the height you get from the non-occluded images. And there's this characteristic excitation, inhibition, excitation temporal profile. But there are differences between the responses to the different images. And again, we can measure the time at which these responses start to diverge across images, and that is around 85 milliseconds after stimulus onset. So there is this delay before the occluded information starts to come into cortex and before it tells you something about what's in the images. Now, of course, the fact that these lines are different doesn't mean there is actually information about what's in the rest of the scene. So to determine if there is information, we used a decoding approach. So here we trained linear discriminant analysis decoders. These are multi-class, so they tell you which one of the 24 images was being presented. And what you see now on the y-axis is the decoding accuracy, and this is done in a temporally resolved way, so over time. Now, for the non-occluded images, the decoders go from chance performance, which is about 4%, all the way up to ceiling performance almost immediately. So they go straight up after about 40 milliseconds. Again, it's not too surprising. There is visual information in the receptive field. We can decode it very easily. For the occluded images, it, again, is slightly delayed. It takes some time, and the decoders only reach ceiling performance at around 100 milliseconds after stimulus onset. But we are able to decode the rest of the scene from the spiking activity in V1. So we already answered our first research question. Yes, the spiking does contain information about the rest of the scene in the occluded region. Now we can dig into that a little bit more deeply, trying to find out what sort of information is being sent back to V1 here by looking at cross decoders. So in pink, we see a decoder that was trained on the occluded data and then tested on the non-occluded data. And you can see that one does reasonably well. It's well above chance. Whereas the non-occluded trained and then tested on the occluded does rather poorly. Now, the reason for this, we think, is that when you train on non-occluded data, there are visual features in the receptive field. You know, there are orientations, there are contrast elements. And that's what the decoder picks up on. These are strongly driving the cell. So when you show an occluded image, those elements aren't there. And apparently what is being fed back to V1 is not the very local individual elements that should be in the receptive field, right? So the decoder fails. Whereas in the pink case, when you train on the occluded data, there are no visual features in the receptive field. So the decoder has to pick up on something else. It picks up on what is being fed back to that region. Now, we don't know exactly what that is, okay, but people have suggested it might be something more like a rough sketch of the image or something related to the gist of the image. But it's something that is also there in the non-occluded state, and so we are able to do some cross-decoding. Now, we can also, just going to skip over that, we can also look at this decoding in a slightly different way by comparing it to what happens in humans. So in human data, we actually can compare the images by putting them into the same framework. So if we look at Tyler's study, he showed these exact same 24 images, and we can compute representational dissimilarity matrices. Right, so I'm sure many of you are familiar with these, but each point in this matrix reflects the dissimilarity between the response to a pair of images across all the voxels in V1. Okay, so dark points would be a pair of images that are very dissimilar in their response across voxels, whereas light squares would be a pair of images which produce a very similar response across voxels. Okay, and that gives you kind of fingerprint about how V1 is representing this occluded information. Now, we can do exactly the same in monkeys, but we can do this in a temporally resolved way, right? We can do this for every single time point in the data because we have very high temporal resolution. And now what we can do is actually compare the monkey and the human data once we've put it into this same framework, right? So we can do a pixel-wise by pixel-wise Spearman's rank correlation between these matrices and see, is the informational structure that's being sent back to monkey and human V1 having the same structure? The result you see on the bottom, and that shows that initially there are no correlations between the monkey data and the human data. That's not surprising, right? Before the stimulus comes on the screen, all the monkey responses are very, very, very similar. But after about 100 milliseconds, you see these correlations reach very high levels. In fact, they're close to the ceiling sort of levels that you might be able to achieve given the noise in the data. So this shows something which I think is kind of remarkable, that what we're seeing here is that despite the difference in species, despite the difference in technique here, the information that is being fed back in the spiking in V1 has very similar information structure to the information that's being fed back and is detectable in the bold signal in humans in V1. Okay, so now I'm going to switch over to a different species to carry on our look into what is being fed back to V1 in the absence of feedforward information. So we're going to turn to mice. In mice, we had head-fixed mice, and we were first doing electrophysiological recordings in V1, but we're using slightly less naturalistic stimuli than the ones we were using before. So these very simple grating stimuli, where we either had stimuli in the receptive field or a uniform stimulus across the whole screen, or an occluded image again. So we have this gray circle which was centered on the receptive field. Now, we were very careful to ensure that the actual occluder was centered on the receptive field and that the 100% of the receptive field was inside the occluded region, right? We don't want any of the background information to get into the receptive field in a feed-forward way. And we could see very clearly that there was no feed-forward activation of the receptive fields in the occluded state. So what you can see here, for instance, in the black and the gray lines are the visual responses caused by the classical stimulation of the receptive field. Whereas in the red line is when we have the occluder. And what you can see is, again, it takes some time before the occluder actually activates the cells. And this first initial burst of spiking that you see in the other conditions simply isn't there. And that was true across the population of cells that we recorded from. Now, we can also see that by looking across the different layers of V1. These were laminar electrodes, and we could look across the superficial layer 4 and deep layers of cortex. That's what you see on the y-axis, across time. And normally, if you show a visual stimulus in the receptive field, this will activate all layers, and you get sustained activity in the superficial and layer 5. But if you look at the occluded information, you can see it's like somebody cut a hole in layer 4, and that there was actually no initial spiking activity at all in that layer. I think you can see that even more clearly if you look at current source density, which shows you the current flow that is going on in cortex. Normally, when you show a visual stimulus, you get this big current sink as the input comes into layer 4 and drives current into neurons. So that's this big red blob that I've arrowed on the slides for the classical stimulation and the uniform stimulation. But when the blanks situation, where we have an occluder, that initial current sink is completely absent. Okay, so here the only point is really that there is no feed-forward activation of the cortical microcolumn using these occluders. And other groups, for instance, the Scanziano group, have shown that if you inhibit the high visual areas in mouse visual cortex, then you can also completely remove these contextual effects. Now, next we were interested in a particular puzzle, which is why is it able to drive spiking activity in visual cortex? We normally think of feedback as being rather a modulatory signal, a weak signal that is coming into the apical dendrites of cells in V1, and only really able to drive cells if it is coincident with feed-forward activity. So if you remove the feed-forward activity, how can you actually drive the activity in using feedback? To look at this, we suspected that what might be happening is that in occluded states, that the cortical microcircuit might be disinhibited. So we started looking at different interneuron subtypes in V1, because there's this well-known disinhibitory circuit within the different interneurons of primary visual cortex. So it's well known that pyramidal cells are the excitatory cells in cortex, and they receive inhibition from parvalbumin interneurons, PV cells, and from somatostatin interneurons, more to the apical dendrites. But interestingly, there is a disinhibitory motif where VIP interneurons actually inhibit somatostatin interneurons. So that prevents the somatostatin interneurons from inhibiting the apical dendrites, and that releases the pyramidal cell from inhibition. So we hypothesized that we might see more activity in VIP interneurons in occluded regions of the scene, which would perhaps allow the cells to become more responsive to feedback input. So here we see the responses to different cell types. So on the left, we have the pyramidal cells, the excitatory cells, then parvalbumin, VIP, and somatostatin. Now, what we saw is that, first of all, if we look at the excitatory cells, they respond very well to uniform stimulation, but also very strongly to these contextual stimuli. Again, it was slightly delayed, but there's still a strong response here. Much more stronger than in the monkeys, perhaps because we used a smaller occluder here. The parvalbumin neurons do pretty much exactly the same thing as the excitatory neurons. But VIP and SST, or somatostatin, do something really quite different. So for VIP neurons, the contextual stimulus is the favorite stimulus of a VIP interneuron. It responds extremely strongly to a contextual stimulus, to a stimulus where there is an occluder. Even more so than it does when there is actually information in its receptive field. In contrast, the occluded image is the least favorite stimulus of a somatostatin neuron. It's actually suppressed when you show an occluded image. So we think this shows strong evidence for a disinhibitory circuit here, where when you show an occluded image, VIP interneurons are strongly enhanced. They suppress somatostatin cells, and that releases the pyramidal cells from inhibition. And that allows them, therefore, to be more sensitive to feedback inputs coming into their apical dendrites, and might allow feedback to actually drive spiking activity. We tested this causally by actually optogenetically inhibiting VIP interneurons. So we put an inhibitory opsin in the VIP cells, GT-ACR2, and when we shine light on cortex, this silenced the VIP interneurons. And what we found is when we did that we were able to reduce the level of this contextual effect, this occluded effect, to about 60% of its previous level. Okay, so it seems therefore that normally VIP interneurons are amplifying up the response to feedback connections. But we couldn't get rid of all of the effects, so perhaps this might be an experimental thing that we couldn't silence all of the VIP activity, or perhaps there are other mechanisms by which feedback can still drive activity even in a non-disinhibited cortical microcircuit. Now, the final thing we wanted to look at was to look at the information that was present in this feedback signal in the mice. So in each of our experiments, we used 6 orientations in the background. And we had many cells which are orientation tuned. So here's an example of a classically orientation tuned cell. This cell likes something around 80-degree orientations. And to analyze this data, we took the orientation tuning of each cell and we shifted it to 0 degrees. So 0 degrees became the preferred orientation of the cell. And then we recoded the data so that into the difference from the preferred orientation. Now, what you can see if you do this is that for uniform stimuli, of course, you get a very, very strong response to the preferred orientation of the cell, which very quickly decays away as you move the orientation away from the preferred orientation. And it comes down to close to spontaneous levels. But for the blank stimulus, for this occluded stimulus, actually it's quite different. It responds reasonably well to all of the different orientations in the background. Right? So it almost doesn't matter which orientation is the background. These cells will all respond. There's only a very subtle orientation tuning left in this curve. So you can probably see the response at 0 is slightly higher than the response to the other orientations. And this was significant, but still rather subtle. So this seems to argue against the idea that feedback is really bringing back the specific feature that should be present in the receptive field here, where we'd expect to see most of the activation at 0 degrees. Instead, it seems to be more of a subtle bias across the population. So that raises the question, could we even decode which orientation was in the background in this particular case? And the answer to that is yes, we can. So we can run these same classifiers again that we used in the monkeys, and they work perfectly well. Okay, so it doesn't matter whether you train on uniform, test on uniform, train on blank, test on blank, every combination gives you above chance decoding. So it seems that although there are only subtle biases in activity, they are enough to drive these decoders and allow you to decode what is present in the background. And these decoders are definitely picking up on orientation-tuned information. If you look at these confusion matrices, where we look at what the classifier chose based on the actual orientation that we tested at, you can see that when it does make a mistake, it tends to make a mistake at a neighboring orientation. Now, interestingly, we could also look at which cells were most informative for these decoders, so which cells carried the most weight in the decoder's decision. And actually there we did find cells, a small population, something like 4 to 5% of cells, which did show very strong orientation selectivity, even when there was nothing in their receptive field. right? So here you see the response of a cell in gray when it is being stimulated by visual information, and in red when the receptor field was blank. And you can see it gives a very strong selective response to the background orientation. So what we suspect might be happening then is that on top of this weak bias that is present in many cells, there is a small subpopulation of cells which does contain selectivity about what should be in the receptor field if it wasn't for the occluder. So to kind of wrap that up into one slide, here we have an occluded region of V1. We have cells which prefer different orientations, and these have different colors in this image. And when you show an occluded image, you will activate all of these cells, right? It doesn't matter what the orientation is in the background. But there's a subtle bias, and the blue cells, probably almost too subtle to see here, the blue cells are going to increase their activity slightly as they prefer the orientation that is in the background. On top of that, we have a small subpopulation of cells, these supercells. We don't know what they are yet, but we're very interested in trying to find out, which do actually strongly respond to the background orientation. Okay, finally, one last thought about what might be happening to the occluder. Actually, I'm going to skip that, I think, in the interest of time. So I'm just going to give you my takeaway messages. So when the images are included in the brain must represent two things at the same time, the occluder and the occluded information. Information about these two things are fed back to V1 and affect spiking activity in this region. The majority of cells in V1 actually represent represent the actual occluder itself, the gray circle. And the strength of this representation is dependent on the perceptual organization of the scene. But the occluded information produces subtle biases in the large majority of cells and larger effects on a smaller subpopulation of cells. So finally, just the acknowledgements. I'd like to thank everyone at the Netherlands Institute for Neuroscience who took part in this. So Paolo, Feng, and Singh, who did the monkey work, Lisa, Sridip, and Peter, who did the mouse work, and the team at the University of Glasgow, Tyler, Lucy, and Lars, for providing us with the human data and for all their discussions about what this might mean. And finally, thank you for listening.